The last part of our blood lecture series will include white blood cells as well as platelets. So white blood cells provide immunity. Another name for white blood cells is leukocytes. Leuko means white. So leukocytes mean white blood cells. And we have two types of leukocytes. We have granulocytes and agranulocytes, basically describing whether or not they have granules within the cytoplasm. So granulocytes do have cytoplasmic granules. There are three subtypes of granulocytes. There are neutrophils, eosinophils, and basophils, basically describing how the cell stains. For agranulocytes, A means without, so agranulocytes do not have any cytoplasmic granules, and there are two subtypes of agranulocytes. There are lymphocytes and monocytes. Now you can compare the relative size of the different lymphocytes by using coins. For example, the largest of these cells are monocytes. Monocytes, we can compare them to, they're not the size of a quarter, but when comparing other cells, they would be, you would consider them a quarter, whereas the smallest of these cells is a lymphocyte, and relatively speaking, we can compare lymphocytes to monocytes by saying the lymphocyte would be the dime of the bunch. So the dime being the smallest coin within that bunch. And then granulocytes, we can compare them to pennies in that bunch. So, or not pennies, nickels, I'm sorry. So lymphocytes being the smallest, being the dime in this group of cells, with monocytes being the largest or being the quarter. Relative to the rest of these cells. So first we're going to talk about granulocytes, and the first granulocyte we'll talk about are neutrophils. So neutrophils are the most numerous of all the leukocytes, make up about 60 to 70% of all white blood cells. Now, physical characteristics, they have neutral staining granules, and they do have this multi-lobed nucleus. If you ever take a histology course or even anatomy, you'll see a neutrophil within a blood smear, and sometimes it looks like a cluster of kind of grapes that are interconnected by these little lines. But they do have a multi-lobed nucleus and neutral staining granules within the cytoplasm. Now, we talked about how within the bone marrow, white blood cell production makes up about 75%, and that's because neutrophils, for example, have a very short lifespan. Within the body, neutrophils have a half-life of about 6 hours. However, in active infection, they are the first responders and will attack a pathogen, and then also can be destroyed in the process. So they can have a lifespan lasting anywhere from seconds to minutes in the case of active infection. Now, neutrophils are one of many leukocytes that have the capability of what's called diapedesis. So dia means across, ped means foot. Basically, they can find an opening within the capillaries and squeeze through the lining of the cells of the capillaries to get to the infection within the tissue. So they leave the blood, so neutrophils leave blood to enter tissues to go to the site of infection to help fight off whatever pathogen is causing that infection. Functions of neutrophils include phagocytosis. So phagocytosis, think of the Pac-Man cells. So Pac-Man cells can engulf and destroy other cells. Mostly they phagocytize bacteria. If we have an increased number of neutrophils, usually this is a good indication of the presence of a bacterial infection. The next leukocyte or granulocyte that we are going to talk about are eosinophils. Now, eosinophils make up 2 to 4% of all white blood cells. They have very distinctive physical characteristics. For example, their granules within the cytoplasm stain a reddish color. So eosin stands for reddish, so they have reddish staining granules within the cytoplasm. Eosinophils have a bilobed nucleus, so meaning there are two lobes, or what the appearance of two lobes to the nucleus. Also, they are capable of diapedesis. Again, leaving the blood to enter tissues to fight off infection. Now, when I think of eosinophils, I mostly think of parasitic infections. So eosinophils are predominant in parasitic infections. They also help protect against hypersensitivity reactions, so hypersensitivity or allergic reactions, by releasing antihistamines. Now, when we do a cell differentiation or cell count, a CBC, if we can see a high eosinophil count, usually we can suspect the patient has a parasitic infection. So with regards to protecting against allergic reactions by releasing antihistamines, they block the release of histamine. Now, histamine is what causes the allergic response, causes the inflammation, and all the things that you'll see with an inflammatory response. When we think of antihistamines, we think of medications that we can get over the counter, such as Benadryl or Claritin. So these medications basically block the release of histamine. And again, an increased number of eosinophils usually is indicative of a parasitic infection. So the third granulocyte that we'll talk about are basophils. They are very rare. Usually you don't see them in a blood smear, and that's a good thing. They make up about 0 to 1% of white blood cells. They have very dark purple staining granules within the cytoplasm. When I describe a basophil to my students, I think of a chocolate chip cookie cell. So it looks like a very dark chocolate chip cookie. You won't be able to see it, but basophils also have an S-shaped or bilobe nucleus, and it is occluded, meaning you can't really see it behind the dark staining purple granules within the cytoplasm. Now, basophils are unique in which they do not undergo diapedesis. They don't undergo diapedesis because they basically are functionally similar to mast cells. So basophils do not have to leave the bloodstream. These cells will go to the area of inflammation by traveling within the blood vessels. So they'll go to the localized area of inflammation, and then they will help with that inflammatory response. So functions of basophils, they will enhance the allergic or inflammatory response. So for example, the responses that are typical of an inflammatory response include heat, pain, or redness in color. So for example, basophils will release histamine. Histamine causes vasodilation, so blood vessels will dilate, providing a red color. Now, this is helpful in that we want to have more blood flowing to the area of inflammation so that we get the proper nutrients going to those areas of inflammation, so that the cells that do undergo diapedesis can exit the blood vessels and go to the tissue where the injury is occurring. Basophils also reduce serotonin. Serotonin increases capillary permeability, again, allowing for the cells that can undergo diapedesis to leave the blood and go to the tissues. Basophils release heparin. Heparin is an anticoagulant. So not so much that you are having a large amount of blood loss, but to the effect where a small amount of heparin will increase blood flow. Again, increasing blood flow to the area of inflammation or injury. The next type of leukocytes that we're going to talk about are the agranulocytes. So A meaning without. These are leukocytes that do not have cytoplasmic granules. The first one we're going to talk about are monocytes. Monocytes make up about 3 to 8% of all white blood cells. Now, these are the largest of the leukocytes. They're large, they're pale, no cytoplasmic granules. They have a distinctive bean-shaped or kidney bean-shaped nucleus. These types of cells, or these monocytes, are produced in lymph as well as bone marrow. So when monocytes are produced in bone marrow, they are said to be myeloid in origin. And if they're produced in lymph, they are said to be lymphoid monocytes, or lymphoid in origin. So macrophages are actually the strongest and longest lasting of the leukocytes, and they are the largest. Again, they are the quarter coin compared to the other leukocytes. These cells are capable of diapedesis, meaning they can enter tissue from the blood. When they enter tissue, they become macrophages, and macrophages, of course, have that phagocytizing property, meaning they are allowed to they can eat either antigens or other cells. Monocytes are important in that they fight off infection, and while they are more stronger and more effective than a neutrophil, they are a bit slower. So when a monocyte enters a tissue, it becomes a macrophage, but they can be called other cells. For example, if a monocyte enters bone tissue, it is called an osteoclast. An osteoclast is a cell we know that absorbs and helps break down bone. If we have an increase in number of monocytes, this is usually an indication of a chronic, meaning a long-lasting type of infection that is occurring within the body. The next agranulocyte are lymphocytes. So we know that lymphocytes produce antibodies and also help enable immunomemory. So lymphocytes make up about 20 to 25% of all white blood cells. Physically speaking, physical characteristics, they are the smallest of all the white blood cells. They are the dime when you think of coins relative to the other cells. They have a very large dark staining nucleus. When I describe these cells to my students, I usually talk about lymphocytes having a fried egg appearance. So a very large nucleus with a very small rim of cytoplasm at its edges. So lymphocytes, like monocytes, are also produced in lymph and bone marrow. Again, they're lymphoid if they come from lymph, and myeloid if they come from bone marrow. So lymphocytes have different subtypes. We have T lymphocytes, or T cells. T cells are lymphocytes that mature in the thymus, thymus being a very important lymph organ. B cells, or B lymphocytes, they originate and mature in bone marrow. Now, these cells are also capable of diapedesis, meaning they exit the bloodstream and enter tissues. So we know that they have specific functions. We're not going to get into which specific cells have specific functions, but generally speaking, lymphocytes do make antibodies, and we know that antibodies can fight off specific infections, specific antigens. Lymphocytes also have immunomemory, meaning if you were infected with a specific pathogen in the past and then are reinfected, lymphocytes have memories to help fight off any subsequent infections with that same pathogen. So lymphocytes are responsible for making antibodies to fight specific infections. This is how we create vaccinations. So hopefully one day when we have a vaccination for coronavirus, we can use this vaccination to help fight future infections. And again, immunomemory, if our body is attacked by a pathogen and then have another attack by the same pathogen down the road, memory cells can fight off that specific pathogen. Lymphocytes are responsible for organ rejection, as with transplant of organs. So if we have a transplant of organs from a donor, we want to make sure that the donor is as closely matched to the recipient as possible, because if not, lymphocytes can attack that organ from the donor. And this increases organ rejection within the recipient. So this is just a nice summary, a table of all the different types of leukocytes. We have the granulocytes, the neutrophils, eosinophils, and basophils. And then we have agranulocytes being the monocytes and lymphocytes. And I'm pretty sure I told you guys about this mnemonic earlier in the semester. The mnemonic will help you remember the prevalence of the leukocytes. So never let monkeys eat bananas. N standing for neutrophils, neutrophils being the most prevalent of the leukocytes, and then next are the lymphocytes, then the monocytes, eosinophils, and finally the basophils being the least prevalent. So I would definitely take a look at this table to help you study the different types of leukocytes as well as some of their general characteristics. Next cell we're going to talk about are platelets. So platelets are also known as thrombocytes, thrombo meaning to clot, so these are clotting cells. Thrombocytes are actually not cells, rather they're fragments of cells called megakaryocytes, and these megakaryocytes will mature in the presence of the hormone thrombopoietin. So here we see the megakaryocyte and then fragments breaking off as platelets into the bloodstream. So again, platelets are small fragments of megakaryocytes. Numbers are usually anywhere between 200,000 to 400,000 cells per microliter or cubic centimeter. These cells are anucleates, meaning they don't have a nucleus, but they do contain endoplasmic reticulum. And this is important because thrombocytes are able to produce chemicals that aid in clotting. So clotting can be activated. These cells are activated or inactivated by chemical signals. So the main function of the thrombocytes is clotting, and basically they act like Velcro particles. They can stick together and aggregate to help form a clot. So this clot, of course, helps stop bleeding if there's damage to a blood vessel. Now, clinically speaking, we take a look at the differentials for thrombocytes, especially in certain conditions where viruses can attack thrombocytes. For example, I talked about dengue fever. Dengue fever is very prominent in the Philippines. It's a virus whose vector is the mosquito. And during the rainy season, there are lots of mosquitoes because a lot of water that is stagnant allows for proliferation of a lot of mosquitoes. So mosquitoes carry the dengue virus. Patients come down with dengue fever after they've been bitten by a mosquito, and usually this virus will attack platelets. I've had one patient who came down with dengue fever, and we were very concerned because his platelet number was 5. Not 5,000, but 5. So he was in very increased risk of bleeding out. So all we could do is basically provide supportive treatment, lots of fluids, and making sure that this patient doesn't bleed out, control the fever, that sort of thing. But again, we look at this number to help us determine whether or not thrombocytes are present within the patient to help them properly clot if there is injury to the tissue. So the last slide is a summary of the components that we talked about with blood and all the characteristics of blood. So we talked about in our first lecture about plasma, and then we talked about the formed elements that made up about 40% of blood. This is a really important table. Make sure you guys study this table. You could read it on your own. And it will be important when we do group problems towards the end of this section. Now, we know that blood is one of the 3 components of the cardiovascular system. The 3 parts of the cardiovascular system include the heart, blood, and vessels. However, even though we only focus on 2 components, the blood and the heart, in this section, know that there are 3 very important components to the cardiovascular system. Okay, so that is our lecture, and that completes our lecture on blood.